Hello, everybody, and welcome to the latest installment of the Hanson Wade Engage webinars hosted by Corning Life Sciences, Gene Therapy 2.0, Leveraging Transgene Cassette Engineering. This is an online event designed to tackle the current viral vector manufacturing bottlenecks, including immunogenicity, toxicity, safety, and efficacy. To briefly introduce myself, I'm Noemi, a member of the Hanson Wade team, and I will be chairing today's session. The webinar today will consist of two presentations, followed by a live Q&A where you will be able to ask your present questions to the speakers. Please remember to submit all your questions via the Questions tab at the bottom of your webinar dashboard. You can submit this at any time throughout the presentations. We will try to answer all questions in the allotted time, but all unaddressed questions will be passed along to Corning Life Sciences so they can follow up with answers directly. So with that said, and with no further ado, it is my pleasure to welcome our first speaker for today, Gabriela Denning, Vice President of Research and Development at Expression Therapeutics. Dr. Denning has over 16 years of preclinical development experience in the cell and gene therapy industry with a focus on lentiviral-based gene therapies. Her experience spans preclinical development to IND-enabling studies and has led various successful regulatory submissions for first-in-human clinical testing. Dr. Denning has served various roles at Expression Therapeutics and currently oversees all research and development efforts in bleeding disorders and oncology programs. She also serves as liaison between cross-functional teams from preclinical R&D, manufacturing, regulatory, and clinical to support Expression Therapeutics pipeline development. So Gabriela, please join me on stage now. Perfect. Welcome, Gabriela. If you can go ahead and share your screen for us.
All right.
Perfect. There you go. Your screen is all good. So I'll see you at the end of your presentation.
Thank you, Naomi. And I want to take the opportunity to thank Hanson Wade and Corning for putting this presentation together. Today, I wanted to talk about how, sorry, I got out of presentation mode, how Expression Therapeutics is really leveraging innovative technologies in transgene cassette engineering and manufacturing to really improve and translate our therapies into the next generation, what we call Gene Therapy 2.0, which is the subject. And the goal is really to improve the safety, efficacy, and the manufacturing costs related to viral vector-based gene therapies. First, I wanted to introduce Expression Therapeutics. Expression is a fully integrated cell and gene therapy company. We really use a suite of proprietary platforms as well as world-class in-house manufacturing to kind of take our therapeutics to best-in-class as possible. We have a research arm, which is where I'm located. It's in Atlanta, Georgia. And then we also have a manufacturing, Expression Manufacturing, which is located in Cincinnati, Ohio. And here, we illustrate how we drive our technologies based on a suite of proprietary platforms in expression cassette optimization, lentiviral vector, as well as cell manufacturing to really be leaders in bleeding disorders and in oncology space. And then we do this by using our manufacturing team to really improve the outcomes and manufacturability of our therapies. So, we've seen over the past decade a tremendous growth in, cell and gene therapy, and that has really culminated with the approval of various therapies that are marketed now in various disease conditions, from oncology, hematology, among others. And it is expected that by 2025, the US FDA will approve anywhere from 10 to 12 gene therapies products per year. Statistically, we know that the majority of these gene therapies under development are mostly focused on viral-based vectors. All these, from the data that we've gathered so far, we know that a lot of these therapies possess dose-limiting toxicities and also manufacturing inefficiencies. And this really leads to low therapeutic potency and high cost of these products. So when we think about the lessons learned from the first generation of gene therapies, there's a lot of improvements that need to be made in the vector manufacture, the way we deliver these therapies, as well as the transgene expression. So if we were to highlight some of the challenges that remain in order to translate what we've learned so far we can sort of put them, and this is specifically talking about viral-based gene therapies, we can talk about three things that we need to really improve on. And I kind of put this in the order that at least expression is really targeting these. That is the control of the transgene expression, limiting the dose-related toxicities of the therapies. And an aspect that I will not touch today much is the immune system. But there's a huge interplay between these aspects of gene therapies that remain challenges in order to really, realize the full potential that these gene therapies have. So when we get a little deeper into what kind of aspects we've learned so far, is that we know that from the long-term clinical data that we've seen so far, is that we have suboptimal vector potency, and of course that's a relationship between the actual transgene cassette and the need to really use high vector in order to improve the clinical outcomes. We know this leads to high MOI usage and potential concerns with the vector copy number. We also have to use strong ubiquitous promoters in order to get to that maximal expression. There's definitely durability of gene expression remains a question. Some of the long-term clinical data, depending on the viral system that you use, has highlighted that this is potentially not applicable to all disease conditions. And because we have the need to utilize, high vector, this leads to safety concerns. And notwithstanding, there's a lot of manufacturing inefficiencies. And also, we're improving in our way we characterize these vectors to have more process control. As we translate these knowledge to the next generation gene therapy, we're really in the need to really focus on improving the vector potency. And here at Expression, we really believe that will really lead to mitigating some of the concerns from the first generation clinical data. This will help to decrease the vector utilization and improve some of the safety profiles. We also believe that by using target-specific expression and using more specific target promoters, you can really control better transgene expression that will lead to durable expression. And notwithstanding, Expression is investing a lot of time in improving manufacturability, which is critical to improving these gene therapies. This is kind of to highlight that our scientific team has made a lot of scientific breakthroughs that really focus on the optimizing transgene and protein bioengineering to develop a best-in-class therapies. And we've also taken the approach of trying to improve other aspects of gene therapy, such as conditioning, as well as trying to improve transduction efficiencies and having a better control. And we're also exploring, in this case, microfluidics. But a lot of the technologies that Expression has been using has been based on these scientific breakthroughs. So this is kind of introducing sort of how Expression is really using a guided pipeline of technologies to improve our gene therapy. I'll start with talking about expression optimization, expression cassette optimization, or ECO. And I want to highlight that a colleague of mine, Harrison Brown, who's the director of computational biology at Expression, gave us a seminar on this, and he really dove deep into this. So I think these slides will be shared, and you can, I encourage you to visit that to get a deeper dive. But just to introduce it here, it's a proprietary codon optimization algorithm. It's an AI-driven technology that really takes advantage of several parameters. We're only not trying to optimize the transgene itself for expression, but we're also taking into context what delivery we're utilizing, whether it's mRNA, AAV, or lentiviral, and also doing mRNA sculpting to really, adapt to the context of use for that therapy. So this is, and I'll get a little bit more into this, but it's really, probably the next generation of what we believe will be important in optimizing transgene cassettes. We also have a proprietary third-generation lentiviral packaging system that we call Lenti-ET. This system really tests a lot of safety features that we've incorporated, and also we've included some of our eco technology to really optimize the potential of this LV system, and it's really fully integrated into our manufacturing arm. And then last, just as a great lead to our GMP manufacturing facility, which really has a tremendous expertise and also acts as a CDMO. They've been working with Corning to optimize lentiviral vector manufacturing, and we also have expertise in manufacturing autologous and allogeneic cell products to converge with our viral vector gene therapy. So diving a little deeper into ECO, as I said, ECO is an AI-driven algorithm. It really tries to customize the therapeutic transgene and how it's going to be delivered in order to mitigate, very specific, target effects that you're looking for enhanced expression, and that's really key to increasing vector potency. So it utilizes bioinformatics and machine learning to really predict optimized transgene sequences and this transgene cassette. So it does this in a context aware, so it's really user guided. You can input the parameters that you wish to inform as it does this iterative process. For example, the target tissue, the type of system, the delivery that you're going to use, whether it's AAV or LV, and also some other features that are important that might be important to you, such as the minimization of open reading frames or the stabilization of the mRNA product that lead to optimized sequences. So as you can see, it's really a multidimensional sequence optimization, and once this process goes iteratively, you select these top eco-optimized sequences, and then you can proceed with your in vitro and in vivo studies to derive your top gene therapy candidates. Here is just to highlight how ECO works and provide some data on this. And we've applied this technology to coagulation factors for increased potency and expression. In this case, I'm showing examples for both factor VIII and factor IX. And in this case, we optimized these sequences for liver-directed AAV-driven gene therapy application. And what we're showing here is data to highlight the increased potency in matched tissue, which in this case is HepG2 cells, versus mismatched tissue, which is baby hamster kidney cells, baby BHK cells. And we're measuring factor VIII activity in the case of plasmid transfected setting. And what you can see is that you can derive a significant increase in both factor VIII and factor IX activity compared to just the general, human codon optimization algorithms that we're mostly familiar with. And this is really not seen in the mismatched tissue. What's really more impressive is the effects of ECO in vivo. And this is something that we early on realized that even though the data in vitro helps guide the selection of these transgenes, truly the impact of ECO is very well highlighted in vivo, which makes sense because a lot of the algorithm is based on this type of tissue analysis. So here in the right, we're showing factor VIII activity, and here is just demonstrating factor VIII exclusively. So here's hydrodynamic injection of plasmid DNA into hemophilia A mice, and you can see a significant impact of the liver optimization, codon optimization for factor VIII activity. And here is actually the actual, in vivo gene delivery process using AAV-based gene delivery, and you can see that you have significant and sustained increases in factor VIII expression compared to the human general codon optimization. So getting a little bit into our third generation lentiviral packaging system, which we call Lenti-ET, we've designed this to have an improved safety profile through the use of low homology backbones, and we also inserted 3 times insulators to avoid translational read-throughs. how we apply the ECO technology to this is to enhance the expression of the accessory proteins, in this case, VSVG and REV. And with the codon optimization for this was done for the actual 293 cells, which are the producer cells, to lead to an increase in vector production. So diving deeper into Expression's therapeutic pipelines and how we apply these technologies to it, we're developing novel gene therapies for both bleeding disorders and in the oncology space using this suite of technologies. In the bleeding disorder space, our pipeline has been mostly focused on the hemophilia A and using ex vivo hematopoietic stem cell based gene delivery. And we're trying to develop a next generation eco factor VIII therapeutics transgene. We are in phase 1 clinical trial, and I'll talk a little bit more about that and where we stand. And although we'll not discuss our oncology pipeline, we are developing, first-in-class therapeutics for both solid and hematologic malignancies. And we're doing these by using a combination of allogeneic cells based on gamma delta platform technology. We call these cells ADECT, for allogeneic donor ex vivo expanded and programmed T cells. And these are genetically modified to express by specific antibodies that are eco-optimized that we call STAR, secreted T cell actuators. So I'm not really going to focus on this aspect of our work, but kind of highlight more on our bleeding disorder space. So diving deeper into these programs, I'm really going to focus a lot on the ET3 program, which is our hemophilia A program, and also highlight some of the bioengineering combined with our eco technology for the hemophilia B, which is the ET9 program. So just a little background. I suspect that a lot of our listeners today might have some experience in hemophilia. So I'll just quickly go through this, but for those of you that are not familiar with this disease, it's an X-linked genetic disorder. It is at least a missing or defective factor VIII clotting protein. It affects 1 in 4,000 to 1 in 5,000 males. And it really leads to bleeding into the joints and muscles, pain and joint damage, and a significant lower life expectancy. Some of the current treatment is based on intravenous infusion of factor VIII products, bypassing agents, and mimetics. And we're familiar with some of the issues with these treatments. It's invasive, they're lifelong. It can lead to inhibitor formation. And there's a lot of, hemophilia has been like the holy grail of gene therapy for many years, and we're very close to realizing this reality. But there are multiple gene therapies under development at the moment, mostly AAV based. And of course, Expression is a player in this field. So diving deeper into our ET3 program, Expression is really taking a very distinctive approach for hemophilia A gene therapy, as we're using ex vivo hematopoietic stem cell and lentiviral-based gene therapy. And the majority of the clinical trials for hemophilia A are AAV-based. And there's long-term clinical and safety and efficacy data. And what that has really demonstrated is that this technology has shown lower vector potency than expected, as well as durability. There's also some immunogenicity concerns towards the in vivo AAV-based treatment, and there's also a narrower patient population because you really have to exclude patients that have seropositivity to the AAV. Expression is using a lentiviral-based approach, an ex vivo lentiviral-based approach, which really mitigates this narrower patient population. And we're using a factor VIII transgene cassette that has increased vector potency. Therefore, it's because of the ex vivo approach, there's the potential for long-term durability of this. And as I said, we can include a broader patient population, including children and inhibitor patients, which really broadens the technology to hemophilia. Diving deeper a little bit into the transgene itself, which we call ET3, it's a bioengineered factor VIII that's been designed for high expression and low toxicity. And it really, the history of understanding how ET3 was derived comes from discoveries made by one of our co-founders, Pete Lawler, who made discoveries in the porcine factor VIII and showed that it was 10 to 100 times higher expression compared to human. This product is now marketed as Ovisir. And another of our co-founders, Dr. Chris Doran, tried to spend years understanding what are the components in porcine factor VIII that make it such a high expressor. And these aspects were incorporated into human factor VIII to derive our ET3 high expressor transgene. We have spent multiple years really understanding the biochemistry, pharmacology of this protein, and we've demonstrated that it has enhanced biosynthesis, greater stability, and indistinguishable immunogenicity compared to human factor VIII. So talking a little bit more about our drug product, we call it CD68 ET3LV CD34 cells. This is a drug product. It is based on using a third-generation cell lentiviral vector that has the ET3 transgene driven by the human CD68 internal promoter. This, when transduced into hematopoietic stem cells, specifically autologous CD34 cells, it leads to expression of the monocyte macrophage lineage for factor VIII production. In our first generation, we're using a non-codon optimized, and then we're now using our ECO technology, and we have derived candidates that have 10 times increased potency. And this preclinical package has been published, and I direct you to our publications, if I'm not going to go really deep into some of our preclinical data. Using this drug product, there are 2 clinical trials that are opened. One sponsored by Expression Therapeutics, and the site is Emory University. And there's another trial in India sponsored by Christian Medical College. This right side really highlights some of the patient participant journey when we enroll patients, and sort of highlights to them, what the treatment will look like. I want to highlight that we have treated in the CMC Valor trial 1 participant, and there's been clinical outcomes now for 5 months, and we expect to share this more detailed information early on in 2023. But I can say that this participant has clinical significant levels of factor VIII. They're no longer require factor VIII infusion, and their safety profile after 5 months is really well. There's a second patient that has been enrolled in the trial and is expected to be treated right before the end of this year. So we're hoping to share some clinical data. The PI for this trial is Dr. Alok Srivastava, and we look forward to having him share more clinical efficacy from this trial. So expression has really, one of our technologies that we really are good at is bioengineering transgene. And I just wanted to highlight another way that we improve the vector potency and transgene potency by using ASR, which is ancestral sequence reconstruction technology. I'm just briefly going to show it because this technology has been applied to the ET9, the factor IX program. And this technology really takes advantage of evolution to guide protein engineering for improved pharmacological properties of your transgene. And this really takes advantage to our current knowledge of the current multiple sequences that are available, and you assemble this to generate a multiple sequence alignment and develop and reconstruct a phylogenetics tree of extant sequences. And if you look at these sequences, you can select possible candidates that have potentially improved pharmacological properties. We can combine this with our ECO optimization technology in order to derive gene therapy candidates through, putting through these candidates through in vitro and in vivo studies. So, using this technology, we've applied this to the factor IX transgene. This has been published, so I'm not really going to go into detail, but we have derived a ancestral 96, this is what we call it, candidate. And again, just walking through how this process works for the bioengineering, you develop this factor IX phylogenetic tree that is predicted. Several of these sequences were de novo generated in vitro and in vivo studies, identify several potential ancestral candidates. There's extensive pharmacological and gene therapy studies to derive this candidate. And what we've shown from using this technology combined with ECO is that we developed a very potent first-in-class factor IX transgene. And in this case, a lot of the studies were done with using AAV-mediated gene therapy technology. And what's amazing about this ancestral candidate for factor IX is that it has independent and equal to activity to the Padua mutant, but it's also additive to the Padua, which means the mechanism is independent of that. And we continue to examine this factor IX molecule. But as a result, at least in the preclinical validation in mice, is that this leads to increased potencies with less vector utilization, mitigating some of the gene therapy related toxicities related to AAV. So finally, I wanted to introduce our manufacturing arm, Expression Manufacturing. During the pandemic, we built a 43,000 square foot facility in Cincinnati, Ohio. And it really is a state-of-the-art facility with expertise in viral vector manufacturing as well as cell processing. It has the capability to scale up to commercial manufacturing scales. And it serves to not only accelerate our internal programs, but to also act as a CDMO. The manufacturing team is led by my colleague, Bill Swaney. He and his team has a tremendous manufacturing experience acting as CDMO at GMP level. He manufactured our product for the phase 1 clinical trial for hemophilia. They have experience in not only lentiviral vector, but also retroviral and AAV. They also have experience in cell manufacturing and have led several GMP campaigns for our gamma delta cell technology. And our lentiviral system is differentiated in that we have this proprietary ECO platform that we built into our technology. We're having a strong partnership with Corning to develop the next generation scalable vector production. We're using the Corning Ascent system, which really takes advantage of using the adherent cell technology, but translating this into what the suspension cell technology is. And I'm not going to talk into detail because my colleague from Corning, Ben, is going to talk about this after me. But this is the PD system that we have in our manufacturing facility, and we're taking, Expression Therapeutics is really invested in taking this technology all the way through GMP scale with our lentiviral vector as well as AAV. So this is really allows for rapid scalability with minimal bridging studies. And look forward to letting Ben discuss this collaboration. So in summary, I hope that I highlighted some of the lessons learned from the generation of clinical data that we have under our belt. And again, in trying to translate gene therapy 1.0 to 2.0, Expression really believes that the best way to address some of these concerns are by controlling transgene expression. And we're doing this by applying not only our ECO platform technology, but by our bioengineering experience to mitigate the dose-related toxicities that viral vector gene therapy has demonstrated. A lot of the toxicities are, very specific to the disease conditions that you're treating. But if you were to find commonalities between these, you would realize that a lot of them had to do with really controlling transgene expression and increasing vector potency. Again, I didn't touch about the immune concerns, but obviously this is something that also is important in trying to improve, and there's a lot of preclinical data out there working on that. So, Expression is using our ECO technology to improve transgene optimization. This is that technology that really takes advantage of tissue-specific codon optimization. We believe this is the next evolution in traditional codon optimization. It leads to greater potency, and also through the combination of transgene bioengineering. And this leads to reduced viral dosing, hopefully potentially mitigating some of the dose-related toxicities by having these stronger, potent viral vectors. In Expression Manufacturing, our manufacturing arm offers the ECO platform to our CDMO customers, and it's really user-guided. So this allows for you to use this tool to optimize and enhance potentially your therapeutic potency. We also offer our LentiET lentiviral system. It's an IP-friendly platform for lentiviral packaging of your transgene if you are using lentiviral vectors. And really our goal is to allow us to guide CDMO customers through, preclinical, that small scale, through all the way through the scalability of GMP manufacturing using the Corning Ascent system. And with that, I'd like to thank you. Here's my contact information, and look forward to hearing Ben's presentation and answering any of your questions. Thank you.
Perfect. Thank you, Gabriela, for this presentation. And now I kindly ask you to leave the stage. But if you have any questions for Gabriela, please submit them using the questions tab at the bottom of your webinar dashboard, and we'll make sure to ask them during the Q&A session afterwards. With that said, it is now my pleasure to welcome on stage our next speaker, Ben Josie, Field Application Scientist at Corning Life Sciences. Ben received his PhD in Pharmaceutical and Biomedical Sciences from the Medical University of South Carolina, working on small molecule and biomaterial-based treatments for cancer and neurodegenerative disease. Ben has held several positions in academia focused on translational research to overcome immunological barriers facing cell therapies. He works extensively with research and process development groups, optimizing assay and cell culture scale-up conditions for viral production and cellular therapeutics. So it is my pleasure now to welcome him on stage to give a presentation on advancing gene therapy innovation through successful collaboration. So Ben, please go ahead now and join me on stage. Perfect. So if you can go ahead and share your screen with us. Yep, there you go. Your screen is up. So I'll let you go ahead and start your presentation, and I'll meet you at the end.
Okay, great. Thanks for the introduction there. And with that, I'll just go ahead and launch right into it. I'd like to thank Gabby for that wonderful presentation that she gave before this, as well as the ongoing collaboration that we've had, which I will now speak a little bit more to about in this next presentation. So this is a bit of a busy slide here, but just to kind of expand a little on what Gabby spoke to earlier about the rapidly expanding space in the gene therapy market, particularly looking at the ones that are viral vector mediated. This is probably one of the most comprehensive graphics that I've been able to find at this point. And without going into too much detail of it, with the new Hemgenix approval, there are now 11 FDA-approved viral vector gene therapies in the United States, with more than 25 currently in late-stage development and over 120 in phase 2 trials, with many more beyond that in the preclinical space. And all of this is really just to say that many of the pain points that we see kind of across the industry, while there are changes in the industry and technological innovations that are going to help streamline and improve this, the sheer volume and demand is creating a lot of pressure on the space. And that is a major contributing factor to a lot of the issues that I'll speak to. So, viral vector manufacturing workflows. There's quite a lot of variation here, as well as proprietary and strategy involved, but they all kind of follow a very similar route. You need to start with a basic material, a cell bank, a viral bank, plasmid DNA, do your quality control testing at this stage. You'll then need to pick a cell culture platform. This is either going to be an adherent cell culture platform or suspension. There's a variety of different cell types currently in the space. I'd say hex are probably the most dominant, but there are some others that also get used as well. And you need to choose a platform system here. And many times over the course of this process, you may need to change platform systems over the course of your process. You'll then move into a virus propagation stage, which depending on your strategy may involve some sort of transfection, transduction, or producer cell. After that, you'll go to a viral harvest, which will involve clarification, treatment, and collection. Purification is a major bottleneck for many in this space. And then after that, you'll move into your final aseptic fill and finish, whether that goes into a vial or a bag, and all of your final QC testing. So there are many unique challenges that are currently facing the space that really demand greater innovation and collaboration to meet a solution. Some of these are process challenges. Gabby mentioned a little bit about how using AI engines and optimizing the vector design can reduce the dosage required. This is a significant benefit to the space because the large numbers of viral vectors and viral particles that are required for the patient doses and the current strategies are really hard to produce. And finding the right upstream production and downstream purification platforms can be challenging, and there are trade-offs depending on choosing one strategy versus another. Greater efficiency and cost of goods in your upstream may result in a higher burden placed on your downstream processes, where you may suffer losses leading to a final lower quality or maybe even a reduced final yield. Supply chain management. This is a huge pain point for people. And a big question for many is whether they should build manufacturing capacity in-house versus outsourcing production to specialized CDMOs. And this goes even a little bit above and beyond just the sheer production aspect of it. This also comes into some of the development work and whether or not you want to license some IP on some already established vectors, whether or not you want to invest in building out your own sort of custom bioreactor designs, or if you want to go with some commercial options, as well as outsourcing some of the quality control testing, and whether or not you want to be the one to develop those assays. High demand in this space, especially in 2022, is a significant burden. Everybody that works in this space, I'm sure, has become familiar with long wait times across the board. And this is really a call out where improved collaboration can be a huge benefit to all sides. Working with your partners and your suppliers to establish accurate forecasting and communicating about this as far in advance, I'd say 6 months to a year at least, are critical to meet some of these timeline demands that the market demands. Now, another one that I'm sure everybody in the space is familiar with would be skilled labor shortages. For all of the wonderful science that everybody in this field is working on, most people, I mean probably all, whether they're working at a small scale or they're moving into a larger scale, they are facing challenges with recruiting, retaining enough people that are talented in the cell and gene therapy, very niche types of skill sets that are required. And there's a lot of innovative and collaborative solutions that are being worked on to address these needs. There's the non-collaborative, just recruitment wars that are ongoing, high turnover rate and job hopping that's going on. And many producers, employers in this space are starting to really invest and look into solutions like automation, as well as collaborating with each other, as well as local academic and educational providers for workforce development programs to try and produce the types of workers with the types of skills that are needed. And across industry collaboration here can really fill the gaps. And this is something that I've seen in many of the clients that reach out to me is they are looking to scale up, and they may have an idea of something along the way that they think they might want to try this, but they don't really want to invest early on the money to hire the knowledge base to make these types of choices. And so collaborations with, whether it's a company like Corning that works in our platforms or other suppliers that's on other platforms, reaching out to these types of vendors and suppliers to use their knowledge base to help you advance at a pace that will meet your needs can have additional benefits related to the labor shortages. And then another kind of elephant in the room would be just the regulatory compliance. This industry is so new and evolving so dynamically that just trying to achieve standardization and realization of what is really important, what needs to be measured, what needs to be reported, and how to do that is a significant hurdle that causes delays for some people. The development of potency assays and the adaptation from the R&D stage of your manufacturing protocols up to the clinical and commercial standards can be extremely time consuming. And so again, this is another arena where reaching out to the appropriate regulatory agencies early on and starting that conversation early on, as well as reaching out to the vendors and suppliers or partners that you might be looking at for your commercial clinical manufacturing, whether that's looking at a supplier of a bioreactor platform or looking at a CDMO that you eventually may want to transfer your R&D process out to. Establishing those sorts of tech transfer conversations early on can really help get around some of the pain points that people encounter down the road. And in terms of the upstream production space, this is where Corning has a relatively significant portfolio and knowledge base to come to bear. There's two broad categories of ways to approach this, and I would classify these as either scaling up, which you see kind of depicted across the top of this slide, or scaling out, which you see depicted across the top of the slide, or scaling up, which you see across the bottom. Scaling out has significant benefits, especially for people who are a little bit earlier on in their process. It provides the benefits of modularity. So if you're not quite sure if you've got any variability in your production needs and you might need to expand or contract your production processes based on a particular demand, or if you're starting off in a normal laboratory sort of T-flask, 2D vessel R&D platform, and you need to get some fast track data. You need to get to that phase 1, maybe even phase 2 level as quickly as possible without having to invest an enormous amount of time or capital expenditures into your process development and your build outs. Tracking into to things like stacked vessels, particularly the Corning HyperStack vessel, it's a very linear process from something like a T-flask. And then if you need to move into a large-scale commercial production of this, you have options in place for manifolding, as well as looking at some automated manipulator options. The scaling up option, on the other hand, involves a bit of a sacrifice in the modularity, but you can theoretically achieve some economies of scale, whether you go through a suspension route and you scale into large suspension bioreactor tanks, which a lot of the biomanufacturing space is historically most familiar with, or if you are working with an adherent cell type, and perhaps you're not interested in investing in adapting that cell type to a suspension platform, or perhaps you've tried to adapt it to a suspension platform and you're just not quite getting the same titers that you were seeing before. There are now adherent bioreactor technologies that can fill these same demands and meet production goals while still working with adherent cells. These are relatively new to the field, and still very much partnerships underway in finding out what the industry needs and achieving and delivering those goals. And the Ascent platform that Gabby referenced a little bit earlier is Corning's offering in this space, which I will speak a little bit more to about. So there's a lot of variation in the viral vector production arena, but kind of generally speaking, people tend to say they get better, superior viral vector yields when using adherent technologies versus in their suspension platforms. This could have something to do with this underlying cell biology. It could have something to do with the increased demand on the downstream processes when you're working with a suspension platform. But overall, in working with our customers, we learned that the stacked vessels, they require a large amount of space. They can become pretty labor intensive when you're operating at large scale. Operator variability and training become a challenge. And there's limited process control here in the terms of online analytics, like monitoring DO and pH. And so our customers really told us that they needed a high yield. They needed it to be scalable from the process development stage all the way into their commercial manufacturing. They don't want to have to change platforms over and over along the way. And they wanted more automation and control, particularly as 2020 and onward have happened. A lot of people this automation function has become more and more of interest to enable a smaller number of workers to produce more, as well as cell line flexibility and analytic controls. And so to meet this need, Corning has developed this Ascent platform. And here in the bottom, you can see just kind of a depiction of some cartoon cells in either a conventional flask, a hyperstack, which would be the most densely packed stacked vessel per footprint available, and then onward to the Ascent mesh kind of zoomed in when you can see the kind of woven structure that would be present in the bioreactor. So how does Corning approach this process? The general Corning innovation process for how we tackle these goals is really collaborative, both within our company as well as with our key clients. It starts off with the inspiration. We speak with our customers that we work with regularly, and we listen to what they have to say. What are their challenges, and what can we do to help them? And then we go through some focused engagements to really understand the critical metrics that are needed and what can be done there. The ideation phase can happen in multiple arenas. We come up with some ideas, we generate some prototypes, and then we'll move on into a series of testing and vetting out these technologies and the applications that they work for until we refine them to something that fills a need. And this is something that I had the pleasure, a process that I had the pleasure of going through rather extensively with the Expression team, both Gabby and Chris, as well as the manufacturing team, Bill and Ninda and everybody over there. And it's a long process, but it is very rewarding. And the end result can be quite impressive. And so a little bit more detail about the Ascent platform. Here, it's an automated, scalable platform for adherent cell-based process designs to deliver surface area intensification, viable cell harvest, and high yield production capacities. It consists of three major components: the controller unit, the vessel, which is essentially a cartridge that houses the packed bed of the bioreactor, as well as the substrate that's packed into that vessel to which the cells adhere for growth. The process development scale unit, which Gabby showed earlier and that you see pictured in the main graphic on this slide, has already been launched. The bioreactors for it come in three sizes: 1 meter squared, 2.5 meters squared, and 5 meters squared. With additional expansion bioreactor designs to come out early next year, and then perhaps a little bit later next year for the pilot and production scales. which will achieve growth surface areas in the bioreactors of up to 100 meters squared and 1,000 meters squared, respectively. Some interesting features for this bioreactor design that make it well suited for viral vector production. If any of you are familiar with chromatography columns, there's a very similar type of fluid dynamics that occur within this chamber, and you get some very uniform residence time distribution. In this slide, it's depicted by an experiment in which a dye was loaded in to the column at the bottom. The fluid path goes from the bottom to the top, and you can see that the band of the dye stayed pretty nicely together and flowed nice and evenly all the way to the top in either the 2.5 meter size as well as the 5 meter size bioreactors. And so why is this really important for the viral vector space? On one hand, it allows for a very uniform cell seeding to occur across the levels of the bioreactor, which we all know is very important for achieving good cell growth. And that's also important from a transfection efficiency stage. Having nice, even, or relatively even confluency across the bioreactor plays a role in your transfection efficiency, with too low or too high confluency cells demonstrating variability. Also having that nice even fluid dynamics. And what we've seen is that this contributes to the ability to achieve very high, upwards of 100%, above 90% transfection efficiencies in HEC-293Ts under a triple transfection PEI-based protocol. So how does this translate into viral vector production? Here in this slide, we see some data looking at an AAV serotype 2 GFP vector production in these same HEC-293Ts that were shown in the previous slide, seeded at 22,000 cells per centimeter squared. In this, we see that the production results achieved are, in the mid E10 per centimeter squared, and the GC per centimeter numbers of the FBR system are comparable to a 2D system. We see very similar results in our lentivirus production as well, in terms of seeing high production efficiency here. Another nice call out here would be for this system is that the cells are capable of being harvested from the bioreactors. So you get nice scalability to allow the incorporation of this bioreactor system into either a seed train, or if you need to harvest some cells off for like end release testing or whatever you might want to take your cells for. And with that, high demand in a rapidly expanding new field is creating cross-industry pain points. And increased cross-industry collaboration is really needed to overcome these challenges within the necessary timelines. These collaborations are leading to the development of new technologies, really specially suited to meet the needs of the cell and gene therapy space. And with that, I'll open the floor to some questions.
Perfect. Thank you, Ben, for your presentation. As mentioned before, we now have a few minutes for the live Q&A session where you'll be able to ask your pressing questions to our wonderful speakers today. So as mentioned, please do submit these questions for Ben and Gabriela by using the questions tab at the bottom of your webinar dashboard. To begin the Q&A session, of course, I'd love to welcome Gabriela back on stage.
Thank you.
Perfect. Lovely. Welcome back. So we do have a few questions from the audience. I'll read them out loud, and if it doesn't say to whom the question is addressed, please feel free to jump in and answer if you think the question is for you. But to start, Gabriela, this question came through while you were presenting. Is your LV system integrating?
It is integrating.
Perfect. And is ECHO only usable for lentiviral gene therapies or also for AAV and AAV gene therapies?
Absolutely. It really could focus first on the transgene and then to the viral system to really adapt to the context of what your gene therapy is. So yes, it's a very flexible platform.
Perfect. And Ben, how do I protect my IP when I'm collaborating with vendors?
That's a really good question. Perhaps Gabby has a little bit to comment on here as well, but I'd say it's really important to think about who you're working with first. Think about the reputation of the company as well as the people engaged that you're going to engage with. And do they have experience in the space? If they do, then it should be a relatively straightforward process to establish an NDA. Do this early on, have this conversation early on, and be willing to revisit it as many times as necessary. It's important for, I think, both parties to realize that, especially in these cross-industry collaborations, there is IP on both sides that needs to be protected. And so as long as there is mutual goals to be achieved, and mutual respect, as well as the proper legal NDA processes are respected as well, I think that there are benefits to both sides to move forward. I don't know if Gabby has a comment on that.
No, I completely agree. I mean, I think that's an important aspect of that collaboration. Obviously, both parties are learning and benefiting from it, but establishing those NDAs and making sure that they protect everybody's interests is important.
Perfect, thank you both. And Ben, focusing now on Ascend, how much media is required?
It's a great question, and very process dependent. I'd say in the viral vector production space, we've gotten some processes down to where they'll run at about 0.1, 0.11 mils per centimeter squared relative to the bioreactor growth surface area. But this is highly dependent on the cells. It's dependent on your transfection, transduction process and what stage in the game you do that, as well as your media formulation. Sometimes less is more, and sometimes it's not. You may be able to streamline a process with a richer media to reduce your media volumes, but you need to look at all your metrics as you go forward there to see if that has an impact on your final yield as well as the quality of the vector you produce.
And can you grow different cell lines?
Absolutely. There's a lot of data presented in this presentation on growing HEC293 cells. We've really optimized this out on multiple different HEC lines, as well as done applications work with cells like Viros, BHK cells, a variety of stem cells as well, and then also some kind of odd, unique cell types. So we haven't tested them all yet, but we've got a nice range that we've already gone through that have been showing some very positive results.
Perfect, sounds great. And Gabby, for hemophilia B, how is your approach different for HEMGenix?
That's a great question, and one that I expected. First, this is a great achievement in the bleeding disorder space to have this approval. What I can say, our data on this Ancestral 96 contract is published. What I can say is that it does not, we have a comparable activity to the Padua mutant. It is an AAV based, the way we have tested this factor 9, and it's also additive. So the mechanism of action is different. It is something that we're understanding more and more through additional preclinical studies. But it's independent of that. So from a patent perspective, it's a different factor 9, and it's also additive. So we see a huge benefit by combining it with the Padua. I hope that answers the question to the audience. But from a gene therapy perspective, it's an AAV-based gene therapy.
Perfect. No, thank you. Just conscious of time that we have 1 minute left. Just wanted to ask that last question that came through. Would you be interested in academic collaborations?
Absolutely. We can. People can reach me or our Chief Scientific Officer, Chris Doran, to discuss any kind of collaboration. And I know that Ben obviously would like that as well.
Absolutely. Do regularly open to academic collaboration. feel free to reach out with any thoughts or questions there.
Perfect. And I just had one last question. It just literally came just through. So do you have any examples of using Echo to improve vector productivity?
So that's something that we're working on for sure. We incorporated the ECO into our accessory proteins, the VSVG and the REV, and we do have data demonstrating some improvements in the output of lentiviral vector. But you know, a lot of the times this type of technology is affected by the transgene that you're trying to make the lentiviral vector for. So it's really, you know, there's dramatic improvement, but it's been validated and tested, but it also depends too on the context of the transgene that you're trying to prove, you know, make in making sure that that doesn't have an effect on lentiviral vector production. But you know, we're really excited about this technology and how it improves lentiviral vector, as well as working with Ben and Corning in trying to optimize the actual production.
Perfect. Sounds great. Unfortunately, we are running out of time. I've seen another question come through. We will be sending all unanswered questions to the speakers so they can follow up directly. But thank you so much, Gabriela. Thank you so much, Ben, for your earlier presentations, for joining me to the for the Q&A session. Please feel free now to leave the stage. And of course, thank you to everyone who took 1 hour of their time today to attend the webinar. Thank you, Ben. I really hope you enjoyed today's session. Please do look out for an email tomorrow from us, as we'd love to hear some of your feedback on today's webinar. If you do have any follow-up questions, any comments, please do email this to myself using the email shown on the screen, info@hansonwade.com, and I will get back to you as soon as possible. But on behalf of Corning Life Sciences and Hanson Wade Engage, thank you so much for your time, and I hope you enjoy the rest of your day.